Thank you for the introduction, Klaus, and thank you for giving me the chance speaking in this seminar series of the PopGen Vienna, a graduate school that I had the pleasure of being part for the last 4 years. During this time, some of you may have already heard about my work. Today, I will focus on a specific project and non-adaptive sequence evolution on Drosophila genome, with an emphasis on the GC-biased gene conversion. Different from my previous presentations, I will take my time to explain what is GC-biased gene conversion, its impact on the genome evolution, and why it's important for the population genetics studies. Starting with a broad definition, gene conversion is a result of the repair of the double-strand breaks during the recombination. And if during this conversion, one allele is predominantly favored over the other, we call it biased gene conversion. To build the foundation, I will dissect each of these terms and provide individual explanations, starting from the recombination. Described here for diploid organisms, we have the sister chromatids formed during the replication, and we have the homologous chromosome, which includes one maternal and paternal chromosomes. Recombination will provide us the exchange of genetic material between these homologous chromosomes. Recombination is initiated by the double-strand break formation, and the ends of this break is degraded to create the long strands of DNA, and one end of this single-stranded DNA will invade the homologous sequence on the uncut chromosomes. Then this intermediate can be resolved via different pathways, double holiday junctions, or synthesis-dependent strand annealing, which can lead to two outcomes, crossover or non-crossover. Crossovers are the reciprocal changes between the chromosomes around this break, but non-crossovers are non-reciprocal ones. This is a simple representation, and it can change depending on the proteins involved, but what is much more important for us, both crossovers and non-crossovers can involve gene conversion. So what is gene conversion? Recombination at all steps includes the formation of a heteroduplex DNA, depicted here with the yellow rectangles. It will include the strands of non-sister chromatids. And if the parental alleles includes some different, for example, alleles, there will be a mismatch. And this mismatch generally fixed through a mismatch repair system. Gene conversion occurs during this repair, and it involves the exchange of like a copying of a stretch of DNA between the non-sister chromatids. And we call the segments of DNA gene conversion tracks. A meiotic gene conversion event leads to non-Mendelian segregation of gametes. What I mean is, here, the blue and red triangles shows the positions of polymorphic site, heterozygote sites. And Mendelian segregation should lead to the 50-50% of the segregation ratio of the alleles. However, in the case of a gene conversion, this ratio distorts. This normally has no consequence at the population level if we don't accept, because if both alleles should have the same probability of the conversion. So at the end, the pool of gametes, on average, will contain the similar amounts of the alleles. However, if there is always a higher probability of one allele to be the donor during the conversion event, this is gonna create a higher allele frequency of this favored allele in your pool of gametes. That is what we call the biased gene conversion. Specifically, let's look at the GC-biased gene conversion. In here, there is a polymorphism between our homologous chromosomes. And after the repair of this double-strand break, the heteroduplexes will involve pairing between the TG and the AC. And during a GC bias gene conversion, these heteroduplexes will be biased towards the fixation of the C and the G. This is one example of polymorphism, but we can have 6 different combinations of our parental alleles, 4 of them being transversions and 2 of them being transitions. But most importantly, depending on the heteroduplexes they will form, we call them as GC changing and GC conservative. GC changing mutations involve heteroduplex pairing between the C and G, which are the strong and the weak bases. And GC conservative involve pairing between the AT or the CG. These are affected by GC bias gene conversion since the repair system has to choose between the AT or CG to fix. That is why they are affected, but these ones are not. There have been experiments revealing this kind of GC bias repair of mismatches, and firstly came from the studies of yeast and the primates. And the strength of this bias was depending on the, for example, the changes are transitions or transversions or the species. As I said, this force has an impact on the population level. And as a population geneticist, we call them, refer them as a non-adaptive directional force. It is directional because it can distort your natural side frequency spectrum like selection. And it is non-adaptive because it favors the CG alleles without an actual fitness consequence. Since our ultimate aim as a population geneticist is to describe the allele dynamics under different forces, its strength is quantified with a composite parameter, big B, scaled with the effective population size. Here, the small b is the conversion bias, and it will depend on the recombination rate, gene conversion track length, and the repair bias in favor of the GC. Well, till now, I have mentioned the molecular mechanisms of this force, but I will take a step back and ask, how are the investigations of the impact of this force onto the genome evolution started? Initiation actually started to understand the feature of the mammalian genome, which are called isochores. Around the 70s, Bernardi's group realized that genomes of the mammalians contain GC content variation over large scales, and they proposed the isochore theory, caricaturized in here. There are variation in GC content, and there are some regions with homogeneous base composition, which we refer them as isochores, and genomes are arranged as the mosaic of this high and the low GC contents, which were transit with a sharp transitions. However, with the genomic data, we started to realize the classic isochore theory does not perfectly fit to the empirical data. Firstly, there was no region fairly homogeneous GC content, but there were some heterogeneity. Also, the clear boundaries between this region, as suggested by the isochore theory, couldn't be defined. So instead of sharp transitions, there were some autocorrelations, which means that the transition from a high GC content to low ones was occurring through an intermediate. The most fitting example to this classical isochore theory was the human MHC locus. But even in there, we were observing heterogeneities, and there were just one sharp transition, but other transitions were mostly occurring more smoothly. So even though there are not isochores, there is still something. There are variation in the GC content, and there are large regions which can be described with a similar base composition. These variations were also thought to reflect some kind of genome organization, since they were affecting all genomic compartments, not just neutral, but also functional. Also, they have been found to correlate it with other features of the genome, like the distribution of the genes or the transposable elements or the recombination rate. That's why it's obvious these variation reflects some kind of components of the genome evolution. That's why there have been a considerable effort to find these genomic segments and explain why there are these variations. In an effort to find these segments, methods, genome segmentation methods have been developed. These methods are purely descriptive. They do not try to determine the cause of this variation, but they just annotate, and these annotations serve as the starting point for your subsequent studies. And one of the prominent methods for this are the hidden Markov models. Imagine you have some data from the genomic windows, and they are belonging to one of the K hidden states that we don't observe. But we have observed data that are emitted conditional on these hidden states. These observed data generally assumed to be drawn some kind of discrete or continuous distribution according to the emission probabilities. Then these hidden states can either stay in the same or they can change to another state according to their transition probabilities. The algorithm, conditional on the transition and emission probabilities, infer the likelihood of your observed data together with the genomic windows or segment of DNA belongs to one of the K hidden states. After this basic description, we also, as a group, developed a HMM method. It's called ordered HMM with emission densities. I will again just give the main assumption of this method. Firstly, we assume continuous emission densities. Specific for the GC content, we assume Gaussian mixture model, which means that the observed data are coming from multiple normal distributions. The second assumption that we think that the data exhibit autocorrelations, which has been observed in the empirical data. To be able to model this, we assume one shared parameters among the hidden states, and there will be state-specific parameter. For the Gaussian distribution, we set the standard deviation as the shared and the means as the state-specific. Then by ordering this state-specific parameter and restricting only transition between the neighboring states, you do model the autocorrelation and you do reduce your number of estimatable parameters. We did apply this to the GC content of the Drosophila genome and inferred the number of hidden states and the parameters of the distribution for each of the states. To be able to define the number of states, there were at least two criteria. First is your model fit, which you can see by the plotting of your likelihood, pointing out to the ideal number of states. In the case of Drosophila, this was 4. Then the second criteria is that whether the means of your distribution for each state was well separated. This was also holding for the Drosophila. These are the proportions of the genomic windows assigned to each of the states. And as you can see, most of the windows were belonging to the intermediate GC content. This is kind of expected because Drosophila exhibit relatively stable base composition. Then we come to do explaining why there are large-scale variations in these genomes. One of the first suggested reasons for such variations was the mutations. However, these models not fully supported by the empirical data, but not rejected until the genomic era. The prediction of this model is that at neutral sites, all mutations should have the same probability of fixation because mutational parameters shouldn't affect this probability. However, there was an observation that contradicts this. GC alleles were having higher probability than the IET alleles, probability of fixation than the IET alleles. Then some people, including the discoverer of this original isochores, suggested that such variation can be caused by the selection. But the problem with this model is that there is no satisfactory explanation for a higher GC content. Since this process is affecting the whole genome, the selection is not acting on the information, but on the GC content. So people suggested why we favor higher GC content because warm-blooded vertebrates, homeotherms, wants them to increase the DNA stability since the GC bases are more thermally stable compared to the AT bases. There were at least two contradictory observations. Firstly, no correlation found between the GC content and the body temperature in the vertebrates. And secondly, isochore-like structures. When I say isochore-like, I mean the GC content variation in the large scales. This kind of structures has been seen way before the origin of the hemiothermy. Given that neither selectionist nor the mutational models provided a satisfactory explanation, focus shifted towards the third hypothesis, which is GC-biased gene conversion. The first strong prediction of this model is that there should be a correlation between the recombination rate and the GC content. Over the years, actually, we collected lots of data that support this prediction, but first ones came from the mammalian genome. In human genome, it has been observed that there are positive correlation between recombination rate and GC content. And there was a specific example from mouse genome. The FXY gene recently translocated to a region with high recombining region. And this translocation has been followed by increasing GC content and increasing substitution rate. The second prediction of this model is that there should be a fixation bias in favor of GC allele, since as I said, this is a directional force. Within species polymorphism data showed that GC alleles segregates at higher frequencies than IET alleles, causing deviations in your site frequency spectrum, and this has been correlating with the recombination rate. The divergence data also showed excess of AT to CG substitutions in all over the genome located in the high recombining regions. These first observations established the GC-biased gene conversion as an important force in genome evolution, and over the years, we started to collect evidence coming from bacterial genomes, unicellular eukaryotes, and multicellular eukaryotes, pointing out maybe the fact that this can be a universal process. And it's worth noting that most of these genomes showing evidence for the GC-biased gene conversion were not displaying isochore-like structure. In fact, the evolution of an isochore-like organization were requiring a couple of factors. Firstly, the strength of GC-biased gene conversion should be strong enough to overcome drift. Also, to create variation, you need variation in the recombination rates. However, the regulation of the recombination rates over years or along the genomes is very different depending on the species. So we cannot expect the same dynamics for each species. I have mentioned the molecular mechanism or its effect on the genome evolution for this force. Now, there is the question why this is important for the population genetic studies. For me, this has twofold explanation because of its relationship with selection and because of its relationship with recombination. So many times I said that this is a directional force. It can distort your side frequency spectra, increase your substitution rates. So when you have the genomic data, it can confound your inference of selection. Also, since it favors the GC allele without the real fitness advantage, it can counteract the natural selection and increase the deleterious mutation load. We also see that this is a recombination-related process, and if we neglect this force, we can actually affect our interpretations about the effect of link selections. All these biased inference of population genetic forces like the link selection or the selection increased, people started to suggest that one needs to extend the null hypothesis of molecular evolution to incorporate the GC biased gene conversion. I hope I convinced you why this force is important for population genetics, and an equally important model organism for population genetics is Drosophila. So one asks naturally whether this force is present in Drosophila or not. This has been long debated, but kind of remained inconclusive. Some studies rejected the presence and said that these are because of the context-dependent mutational patterns. Some other studies suggested that it is present on the Drosophila, but they kind of change in their claims on which chromosomes or species they acting. If you ask me, these studies showing the evidence for GC biased gene conversion kind of do not answer some certain questions which can lead to more strong conclusions. For example, what is its relationship with recombination, or how does it affect our previous population genetic inferences? In one of my projects, I more deeply try to understand the dynamics of this force. But before going into that, I will give some relevant population genetic findings for this species. There is a naturally evolving class in this species, which are the 5' side of short introns, I will call 5SI. And these sites has been used to infer selection or demography. And I want to mention that the length distribution of the introns in Drosophila is very skewed, most of them clustering in the minimal length. So you can find them in almost every regions of the chromosome, making them more valuable as a reference sequence. Furthermore, the first documentation of the effect of link selection has been shown in this species by the positive correlation between the recombination rates and the polymorphism. It has been also suggested that the background selection causes the natural X/A polymorphism ratio higher than the natural expectation of 3/4. The reason for that was higher recombination rate, and X leads to less reduction in the diversity. It's obvious naturally evolving sequences are valuable for a complete understanding of the non-adaptive forces shaping your genome evolution, and they are also valuable as a reference sequence to detect the adaptive forces, but only if you know all these processes that are affecting their evolution. We know these 5' sites showing deviation from the natural expectation. They have an excess of high-frequency GC alleles. So I think some works needs to be done. What is the cause of this force? And in my project, I try to broadly identify and describe the relative contribution of the non-adaptive forces shaping the evolution of these natural sequences. Some questions of the study has been motivated by a recent study of Jackson and Charlesworth. They showed the evidence for the GC favoring directional force in this 5Si sites in the autosomes, but there have been some questions that are not answered. For example, does GC bias gene conversion affects transitions and transversions similarly? Or is GC bias gene conversion also present in the X chromosome? If it is, if the strength of it is similar to autosome or not. Most importantly, our interpretations about the effect of link selection can be caused maybe or affected maybe by the GC bias gene conversion. Do we see such patterns? To be able to answer these questions, our approach was to compare different nucleotide classes along the chromosome, between chromosome, and in relation to recombination rates. For this, we used the multi-allelic model to estimate the scaled mutation rates from the polymorphism data and divergence from the simulants data. I will not go into the details of my methods, but let's look at our mutation classes. These are all the mutations possible and mutation rate matrix. We assume strength symmetric rate matrix, which means that A and T or C and G bases are interchangeable. So these two mutation rates are same, reducing our number of parameters from 12 to 6, and we parameterize them as B, A, B, C, D, E, F. From this mutation rate matrix that we inferred, we can check if there are mutational differences between these nucleotide classes. But apart from the mutational differences, some of these classes might be affected distinctly by other evolutionary processes. As I mentioned, these two classes are GC conservative. They are not affected by GC bias gene conversion, and there is no mutation bias between them, and they are transversions. The other four classes are GC changing mutations, and they do include both transitions, BC, and the transversions, D and E. I also estimated the divergence similarly for the 6 class of nucleotide classes, and I compared the divergence over polymorphism ratio to differentiate between some population genetic forces. How so? Let's look at the under neutral equilibrium condition. The divergence over polymorphism ratio should be T over 2N for autosome, and it should be 4 over 2/3 of the autosome for X chromosome. And by looking at this ratio, I can compare the same mutation classes between different regions, whether there are some deviations or differences. I can also compare different mutation classes. Let's imagine there is a mutation class that is neutrally evolving or under the directional force. With a directional force, I do not expect these ratios to be same, but deviates from each other. However, only with the effect of link selection, this ratio shouldn't be changed because link selection should affect the mutation classes similarly coming from the same region. All this set and summarize, we start with our along chromosome estimates. We did observe the previously known patterns. Divergence does not vary along the chromosome, but polymorphism does. So not the mutation rates, but the effective population size variation creates this, either due to direct or indirect effect of the directional forces. So I do check the divergence over polymorphism ratios and do observe differences between the mutation classes, pointing me out that link selection cannot be the only reason creating this, but there should be a directional force, likely GC bias gene conversion. So GC conservative and changing mutations different from each other, and probably GC changing transitions and transversions are affected differently by this force. Let's move on to the between chromosome. Since we observe there is an effect, something affecting both of these chromosomes, we wanted to check whether the previous interpretation that this higher than expected ratio is because of the background selection. These are the values expected under neutral equilibrium for polymorphism and divergence. But our observed values were high for both of the estimates for polymorphism and divergence. And this cannot be caused by the background selection as suggested before. Firstly, we do observe differences between mutation classes in the polymorphism ratio. And we do observe higher mutation rates for the X chromosome, as we got it from the divergence ratios higher than 1. So, how much of this caused by mutational differences or by the directional forces? In the case of pure mutational effect, the divergence and polymorphism should increase or decrease proportionally for chromosomes. So I do not expect these ratios to change, but stay constant. So I can check this expectation, whether there is a mutational effect or directional force. To check this expectation, I do plot the X over A polymorphism with the adjusted divergence values, which are multiplied by the 3 over 4. If there is an overlap, I can say that the mutation can explain everything, but if there is a deviation, there is an effect of directional force. In this case, expectation holds for the GC conservative mutation, but deviates for the GC changing transition, pointing me out that actually background selection is not causing the differences, but the mutation rate differences can explain the deviation in the X over A polymorphism ratio. And most probably the dynamics of the GC bias gene conversion between autosome and X changing for the GC changing transitions. Then I come to the recombination rate estimates. I divide my data into 4 bins of recombination from high to low before excluding the centromeres and telomeres. And the green ones is my whole chromosome estimates. There is a decrease in estimates with the decrease in recombination. But when I exclude the telomeres and centromeres, this association is relatively diminished. This has been known before, but what is pointing that most of the association between polymorphism and recombination is caused by the differences between telomeric, centromeric, and the central regions. So how much of this association is caused by GC bias gene conversion and the link selection? I again check the divergence over polymorphism ratio. First thing I want to show the association, the variation in the ratios were much more stronger for the GC changing mutation. So it suggests that GC bias gene conversion contributes to this positive correlation. Then I exclude the chromosome ends and check on the central chromosome with the yellow dots. You can observe there is no variation anymore in the ratios for GC conservative mutation, but there is a slight variation for the low recombining region in the GC changing mutations. This shows that link selection is relatively stable in the central chromosome. Also, GC bias gene conversion is relatively stable, maybe a little bit different through the low recombining regions. Since I observed the effect of this force in my diversity patterns, I wanted to see what is the strength of it. So I estimated B from the side frequency spectra data. Given here for the autosome and X, and for the whole chromosome, central and the peripheral regions. The pool data means that I estimated after I combined the autosome and X data. And to be able to check whether the estimates of autosome and X differs from each other or not, I check whether the pool data improve the fit or not. The bold values means that the autosomes and X do not differ significantly. What we got is a weak force around 0.5 for both chromosomes, and they do not differ significantly between autosome and X. I do not want to confuse you, but just make a side note. These estimates are obtained by using all GC changing mutations without differentiating the transitions and the transversions. When I obtain my estimate after I differentiate the transitions and transversions, I do observe slight differences between autosome and X. What was much more important or interesting observation for us, we did observe some increase in strength towards the peripheral region, telomeres and centromeres, which are regions that has been marked with low crossover rates. And this negative correlation was also reflected in the negative relationship between the short introns GC content and the crossover rates. So it's obvious that crossovers and the GC bias gene conversion is not directly related. Some kind of different forces might be affecting that, which might mean non-crossovers. As I told you, non-crossovers also leads to gene conversion events. And unfortunately, we do not have the non-crossover estimates for this species. I do not expect maybe direct relationship between crossover and GC bias gene conversion strength, but I do expect a direct relationship between the strength and the GC content. So I wanted to divide my data into GC by GC content, but obtaining a force that are affecting the GC content after dividing your data according to the GC content might introduce bias. To avoid this dependence, I use the background GC content, and I choose the forefoot degenerate sites for this. This has been justified by the significant and positive correlations between forefoot degenerate sites and the short introns base composition, which is pointing out the common mechanism affecting their composition. So I divided my data into 5 bins of GC content, increasing GC content, and these are our estimate. This is for the whole chromosomes. I do observe a slight increase for the high GC content compared to the low one, even though there is not so much significant difference between the other 4 bases. I can still say that this pattern is pointing out to the effect of GC bias gene conversion, and I was not expecting a very strong correlation since, as I showed before, the GC content of Drosophila is relatively stable. Just to check how was the mutation bias towards AT were changing inside this GC bins, I also estimated the mutation bias for 5 of my GC content bins and for all chromosome, and I did not observe any difference in the mutation bias. From this study, I can draw 2 important conclusions. Firstly, GC bias gene conversion is present in the whole genome, and it differs between mutation classes and a long chromosome, but it is approximately equal in strength between autosome and X chromosome. What is much more important, the presence of this force changes our previous interpretations about the natural sequence evolution, especially about the effect of link selection. Link selection mostly causes differences between the central and the peripheral region, but it is not changing in the central part. Then autosome and X differences is not mostly caused by the link selection, but it is mostly affected by the differences in mutation rates. I think this study adds the Drosophila to the list of species that has been affected by the GC bias gene conversion and giving some credit to the idea of universality of this process. And even though the forces that we inferred was weak, we did see its effect on the diversity pattern. That's why I can suggest that incorporating the GC bias gene conversion into your null model is important when you're trying to infer the adaptive forces, or if you want to get a better picture of the non-adaptive forces other than the GC bias gene conversion, it's wise to focus on the GC conservative mutations. With this, I want to conclude my talk. I thank you also, my supervisor, and all the PopGen Vienna members.